Afternoon, guys. How are you all doing? I haven't played Kahoots in ages. I'm very jealous of you guys organising a Kahoot session. I used to purposefully play my students, and I purposefully lose until the last few minutes and try and get like a hot streak to win. Oh, that's a nice blast from the past anyway. Yeah, how's everyone doing? I've unintentionally as well just gotten rid of my tab. Hello, there we go. Yeah, how are we all? Hello. Home Cram School Club. Yeah, that was the idea. I tried to come up with a witty name, not witty name. Tried to come up with a good name for this. And I thought Home Study Club was a nice one. Anyone else come straight from maths? Oh yeah, you guys have just been with Max, right? What a nice afternoon you get on a Wednesday. Not good. Lockdown is bad. Lockdown is bad. Lockdown is bad. But there are nice parts to it. It's a lovely day, I assume, in most places at the moment. When my web class starts, it won't let me join. Huh. That's weird, Amelia. I don't know what to say about that. What has everyone been up to, apart from Kahoots and apart from, I don't know, not doing very much around the house? What have you been up to? Got the paddling pool out. Nice. Nice. How big is this paddling pool? Is it one of those like ridiculous ones which I've seen people have recently, or is it like a little kid one with like the nice animals painted on the outside of it? Just came from an English Lit Zoom meeting. Nice. Doing some tutoring. Nice. Games. Destiny. Destiny. I definitely played Destiny. I can't remember what it is, though. But nice. Did I see your art piece? Simfuja, were you the one who... Yes, I did see your art piece. Yeah, this is a good point. So I will be doing a I'll give one final account out after today. And I have someone in mind for first place at the moment. So I'm expecting an amazing something or another, but I'll tell you more about it in a second. I did see that. It was very good. Virtual work experience and medicine. Nice. Nothing like a bit of virtual work experience. It's like in between. No animals, but it's not massive. Okay. Oh, I used to like the animal ones. I used to have one of them. Your school decides to do a wellbeing week and set no work. Nice. Nice. My school used to do wellbeing weeks. I think they're a good idea. Oh, it's all very confusing, isn't it? I'm still not too sure what's actually going on. If you guys are going to be going back on the 1st of June, or if you guys are not going back at all, or if you guys are going to go back when your school say, or if you, I don't know, all weird. You signed up, signing up for UCAS. That's exciting. UCAS is like quite an interesting experience. I can't remember enjoying having to write a personal statement, but I don't know, they're interesting. Schoolwork, very nice. Very, very, very nice indeed. Right, I appear not to be lagging today as well. So I've literally done nothing. I've changed nothing, and for some reason it fixed it. So that's good. Phoebe Carter's been running. Right, I'll share my screen because I forgot to do that last time. Okay, you should hopefully be able to see. Ooh, which screen can you guys see at the moment? You should be able to see that one. Excellent. Yes, that's all good. So basically, plan for today then is we're going to be looking at some transport in plants. So I did some transport in animals on Monday. We were looking at, I don't remember what we were looking at. Oh yeah, the heart and stuff like that. So yeah, essentially then, the plan will be, we'll go through some transport and plant stuff, go through some phloem, some xylem, transpiration, translocation, anything that comes up. If you get really confused, feel free to let me know and I'll try and help out. But otherwise, let's get cracking. So before I get properly started as well, this is the one final free ultimate package for one person. Okay, now someone did a really good picture earlier this week, so I've got someone in mind who I think is going to be quite likely to win this. There's also one which there's a big Harry Potter theme. I feel like you guys realize that I like Harry Potter, but you probably don't know anything else about me. So yeah, I've got one free account to give out. All you need to do is follow us on Instagram, post a picture of you doing something like, I don't know, watching the store, watching today, watching now, and basically if you put it on your story and share it, I'll have a look out for the best one, essentially. If you have a private account, which I know some of you will do, then I think you'll need to like send a screenshot of it to us, like DM us, and then we can see that you've actually done it. Okay, but I am going to pick the most creative one, or the most, I don't know, the one that makes me laugh the most, essentially. So that's what I'm looking out for. Just so you know. I've also got a coupon code at the end. Apparently, the coupon code that I gave you last week actually didn't work because I made a mistake, and the person told me to write cardiac, but I wrote cardio because I'm an idiot. So hopefully that doesn't happen again as well. But otherwise, let's get cracking. So which of the following, A to D, is not an adaptation to reduce water loss in plants? So out of these things, what is not an adaptation to reduce water loss in plants? Who won last time? There hasn't been a winner this week, Simfuja. So let's have a look. And I'll answer the question about year 12 applying to uni in 1 second. So which of the following ACD is not an adaptation to reduce water loss? So there is an extensive root system that extends far from a plant, leaves that are reduced to spines to prevent damage from animals, ability to store carbon dioxide, so stomata don't need to open, or surface is covered in reflective hairs. Which of these isn't going to help to reduce water loss? Most of you are going for A. So you guys are spot on. So A is a way to get more water, right? So this is saying water loss. This, in fact, is a way to get water in the first place, right? So it's nothing to do with loss. This is just getting water. Leaves reduced to spines. That's to do with surface area to volume ratio. Ability to store carbon dioxide, nothing to do with it. Surface covered in reflective hairs, nothing really to do with it. I'm glad, Amelia, that you're feeling okay with that. So someone just mentioned, should you apply to uni this year? Apparently 1 in 5 people this year have decided not to apply to university and they're deferring for the year after. So yeah, I think by the time you guys are going to uni, if most of you are in year 12, which I think most of you probably are, I think university would be fine the year after. If you're in year 13 and you're thinking about if you should go next year, I think it's completely up to you. I think things will eventually get back to normal. It's probably we're going to need a vaccine first. But I probably don't want to talk about this too much, just in case I get kicked. So, but we can't travel in the gap year. That is very true. I don't know what that means. Don't know. Did you get some math slides? There's a chance you've been sent some math slides. Hopefully not. Anyway, so, did I actually manage? In fact, there's a chance I just didn't manage to actually get the slides. Hang on. If I just check, because I can send you the slides if not. Does this look like the thing I'm doing? There you go. Just in case you didn't get the slides, I will post them myself. There you go. Anyway, so next one. Which of the following options, A to D, is a reason why plants require specialized transport tissues? So why is it that a plant needs a special transport tissue? So why does it need to have xylem and phloem? Is it to allow osmosis? Is it because they all have a large surface area to volume ratio? Is it to carry sucrose? Or is it to overcome limitations of diffusion? These things are happening. I haven't even said my 2 things. I'm getting way ahead of myself today. So how often does this happen? Twice a week. How long are the sessions? About an hour-ish. And what specs do I do? Basically all of them, but I'm focused on the British ones. Right. So you guys are, again, yeah, you guys are totally correct. Well, it is D. So why is it not A? To allow osmosis to take place? No, that's not why they have a specialized transport tissue. That's nonsense. Because they have a large surface area to volume ratio, if anything, that would mean you wouldn't need one. To carry sucrose? No, they don't just move sucrose, they move water as well. So D. Monday and Wednesday. Although I won't, is next Monday a bank holiday or is it next Friday? Yeah, next Monday is a bank holiday, isn't it? So I won't be doing one next Monday, I don't think. No, is next Monday a bank holiday? Yeah, I swear it is. I think it is. Yeah. Anyway, mistletoe, this one's getting harder then. So I always try and starting easy and getting a bit harder. This is the first one, which is a little harder. So mistletoe is a plant parasite that lives on the stems of other plants. Who knew? Who knew that mistletoe was a parasite? There you go. You've learned something interesting. It survives by removing water and assimilates from the host plant. The mistletoe binds to the stem of the host plant and grows a specialized root-like tissue called a haustorium that attaches to different tissues in the stem. One species of mistletoe, Viscum minimum, contains no chloroplasts. Which of the options A to D explains why V minimum does not need chloroplasts? So the Haustatorium, or however you pronounce this word, I think it is Haustatorium or I don't know, whatever it is. The Haustatorium of V minimum attaches to sieve tube elements. The Haustatorium of V minimum attaches to xylem vessels. The The halcitorium of V minimum attaches to meristem cells. The halcitorium of V minimum attaches to cambium tissue. What do you reckon? Most are going for A. Why sieve tube elements? Why is that relevant? I think it's, you guys have all agreed. This is very unlike you. So yeah, what even is a sieve tube element? Good question, Sarah Mahmood. So a water and a stimulants are flowing. Yes, so good. Yeah, you are right. It is A. So a sieve tube element, in case you haven't come across that before, which all of you should have done if you've done transport in plants, basically they are the cells that make up the phloem, right? So the phloem is made of two cells. It's made of a companion cell, which is on the outside, and it is made up of a sieve tube element. Essentially, what's happening is by having this, whatever it was, the halcitorium thing, having that sort of inserting into the sieve tube element means that anything moving through the phloem, which is a stimulants, so sucrose and amino acids, they will start to move into the mistletoe. Essentially, because it needs water and it also needs sucrose, the only place where it can get both is in the phloem. So the xylem just contains water and minerals, whereas the phloem contains sugar and water, sucrose and water, stimulants and water. And phloem transports sucrose, so no need to make its own food. Yeah, exactly. That's all good. Okay, right. First hard question, I'd say. So figure 19.1, this is a sieve tube element as well. So those of you who haven't heard of that, there you go. It's one of these things. It's got a weird sieve plate in the middle. Figure 19.1 is a diagram that represents the loading of sucrose into the phloem at the source. With reference to figure 19.1, explain the process of loading of sucrose into phloem and its movement in the phloem. So just one little point here. If you do EdXLA, there are some bits of this you don't need to know. Okay, so you don't need to know about hydrogen ions. That's what we're saying. Is international EdXL more similar to EdXLA or EdXLB? I don't actually know. I imagine it'd probably be a bit of both. Resurrection Stone, is there lots of stuff in it about like cystic fibrosis and things like that? So hydrogen ions, Namrata, you've started me off. Hydrogen ions are actively pumped out of the companion cell and diffused back with co-transporter proteins. Oh, very good. So first thing that happens then, in order to get sucrose into my companion cells, where are the hydrogen ions pumped from and to? So they're going from the companion cell. Where are they pumped to? EdXLA, you don't need to know this. I'll tell you how you need to word this instead in a second. No, it's not going into the sieve tube element. This is something that people commonly misunderstand here. So the hydrogen ion isn't getting pumped into the sieve tube element because then sucrose would be going the wrong way. So hydrogen is being pumped out of the companion cell into whatever cell happens to be next door. So it could be like a parenchyma cell, or it could be palisade cell, something like that. AQA do need to be aware of hydrogen here, yes. Okay, so hydrogen is pumped out. So first mark, companion cells. Pump hydrogen ions out, I don't know, into surrounding cells. If I'm saying pump, what do I mean? No, AQA, this is definitely AQA. If you have the CGP book, they ignore hydrogen ions, but the actual official book that's affiliated with the spec has hydrogen. Good, using active transport. So companion cells pump hydrogen out into surrounding cells using active transport. Okay, right. Someone then said, and I'm going to start making this take up a bit less space. We could say a hydrogen concentration gradient is formed. Are the hydrogen ions from water? No. Well, they could feasibly have been at some point, but no. Doesn't really have a name. It's just, well, it does have a kind of name, which I'll tell in a second. Okay, so I'm pumping hydrogen out. I'm forming a concentration gradient. What then happens? It's pumped out to make a concentration gradient. So there's now loads of hydrogen ions here. Sucrose is not pumped in. Good guess, though. That's what Edexcel needs to know. Good. So hydrogen ions get pumped out, and then they basically will move down their concentration gradients, and they take sucrose along, like, with them. So hydrogen concentration gradient formed. Hydrogen ions. Let's not say move. Let's say diffuse. Back in with sucrose or amino acids by co-transport, not con, using co-transport proteins. Okay, so the whole point of pumping hydrogen out is to get a concentration gradient so that it can come back in and take sucrose too, using co-transport proteins. Right, so why do they need to use that process out of interest? What's the point in doing that? Why not just let sucrose, why can't sucrose just diffuse in? What's the point of this? So it could feasibly be against a concentration gradient. That could be it. It could be that there's more sucrose in here than there is out here. That could be one point which you need to overcome. The other thing which is true of diffusion is it only ever gets at best to an equilibrium. Okay, if it can only ever reach equilibrium, it means there will always be sucrose outside that can't actually get in. So this process is useful because it can work against sucrose's concentration gradient, and it means that you can go past the equilibrium point. Okay, yeah, sucrose is also probably too large to just diffuse through anyway. It would need to use facilitated diffusion at the very least. Okay, so sucrose is then going to build up in my companion cell. So I'll start writing over here. Sucrose builds up in companion cells, so it probably would be going against its gradient. And diffuses into sieve tube elements. I'm going to call sieve tube elements STEs from now on. And diffuses into sieve tube elements. Right. How does sucrose actually get transported then? Like 2 or 3 questions, not many. Why is it slanted? I don't know. It just is in this diagram. Don't think there has to be a reason. Increase sucrose in the companion cell, therefore diffuses. Yeah, so it's going to move through. There are actually little holes called plasmodesmata, which it can move through. Is it facilitated diffusion if co-transport proteins used? Yeah, it's a form of facilitated diffusion, but it's actually against the gradient. So you can just call it co-transport. Good. So sucrose is now going to move in here. So sucrose has moved into my sieve tube element, and that has a massive effect on the water potential. Okay, so sucrose moves in. So the water potential of my sieve tube element is going to decrease. Right? So sucrose builds up in companion cells and diffuses into STE. Water potential, which is what this psi symbol is, water potential decreases, which means that water osmoses, which is a word you can use, osmoses in from the xylem, increasing hydrostatic pressure. Okay, so loads of water is going to move in from the xylem. So there's now loads of water. If there's loads of water in there, the pressure is going to build up, as you would expect. It's like pumping your car's tire or your bike's tire up with air. The more you put in, the more pressure there is. Loads of water going in increases the pressure. What actually causes there to be a pressure gradient? Sieve tube element is an STE. So why is there a pressure gradient though? So a pressure gradient assumes that there's a low pressure somewhere else. Why is there a low pressure somewhere else? So you only get movement due to pressure if there is a high pressure somewhere and a low pressure somewhere else. So why is it the case that there's a low pressure elsewhere in the plant? Why does the sink have a low pressure? So not loading of sucrose, you want to say unloading. So yeah, essentially, let's say either this is my sink. So this was my source. The sink, sucrose is going to be moved out by diffusion first, facilitates diffusion, or actually it could be normal diffusion if there's plasma, it doesn't matter. But it'll move out by diffusion and active transport to make sure all of it moves out. If all the sucrose moves out, then the water potential down here will actually be very high. It'll basically be pure water. All the sucrose moves out, there's only water that's left. So the water potential here will get very high. So this means that water will also move back into the xylem. So we want to say, maybe I'll write this like over here or something, or I don't know, I'm running out of room a bit. So unloading at sink lowers water potential. So water osmosis back to xylem. Lowering hydrostatic pressure. Okay, the point of that is a pressure gradient is then established. Which equals mass flow or mass transport. Okay, if you do edXLA, all of this is still true, but instead of saying this bit, just say that sucrose is actively transported into the sieve tube element. Yes, that's what you need to, well, all you need to say. So that's what you need to say to get your 6 marks. I know it's a lot, but this is something you're expected to know very well at A level. And I think it's logical. Like sucrose moves, in fact, this first bit's a bit weird. So this first bit's hard. If you can remember that, well done. But sucrose moves in. That has to lower the water potential. Water will move in. The sink, you know that the sucrose is going to move out, which will increase the water potential. So the water's going to move out again. So, what are you saying? Sucrose is a solid? Yes. Less sucrose in the sinks, therefore, a concentration in water potential? Yes. 6 mark questions are complicated and long-winded. Agreed. I'm glad you like my explanation, though. I don't know if I've got any more questions on this, but yeah, feel free to learn that off by heart. Can you explain how water comes into it, please? Yeah. So, essentially, what's going on, right? If I go onto another page, here's my phloem. Here's my xylem. They're right next to each other in plant. They're literally right next to each other. So as soon as my sucrose moves in, sucrose will dissolve because it is soluble. So that's going to cause the water potential here to massively go down, right? Water potential is a measure of the amount of solutes dissolved in water, essentially. So the water potential is low over here. The xylem, which tends to have quite a high water potential, is therefore going to move water from the xylem to the phloem. When sucrose moves out, the water potential is going to be very high because there's nothing else in it apart from water. Even so high that it's going to be higher than the xylem. So the xylem's water potential comparatively will be lower than the phloem's. So water moves back again. Okay, that's it. What if there ain't no water come into the xylem? Then what would happen? There's always water. There's always going to be water in the xylem. Like unless the plant's dying, and then if a plant doesn't have any water, then it won't be able to do active transport. Well, sorry, then it won't be able to do translocation. The gradient bit is just saying that things can only move by mass transport if there's a pressure gradient. So, for instance, if I've got a toothpaste, that kind of looks like a toothpaste to me. If I just leave my toothpaste like that with the lid off, nothing happens, right? If I put loads of pressure here, then there's a high pressure here. There's a low pressure here. So the inner bits are going to come outwards, right? If I swapped it around, then air would be pushed into my toothpaste. But there's a pressure gradient because there's a high pressure here, low pressure there. That causes movement. Right, cool. Next complicated bit. There's only 2 complicated bits to this, I'd say. So anyway, the cohesion tension theory is often used to explain the mechanism by which water moves up the xylem from the roots to the leaves. Use this theory to explain how water moves from the roots to the leaves. Yeah, plants can transport water even when they're dead because the xylem itself is dead. There's nothing active needed for the xylem to move. It just needs evaporation. So water is pulled through a transpiration stream. Yes, that is true. How though? What's the difference between cohesion and adhesion? Cohesion is when two water molecules stick together. Adhesion is when water sticks to the cell wall. So active transport against concentration, thus cohesion allows water to be pulled up. No, not active transport. Water molecules form hydrogen bonds with each other, which means that they move by cohesion. That is good. So you guys know a little bit about this. What's the first thing that happens? So I always start this off in one place, which seems a bit strange, but I'd always start explaining this from one place. Freya, you are spot on. So the first thing to mention is that water evaporates out of the leaves because of the sun or something. It evaporates out of the leaves due to heat from the sun. Okay, so water evaporates out. So I'll change the color around in a second and show you. In fact, no, let's do it now. What does that do? So when water evaporates out, what does that produce? And you could say it's coming out of the stomata as well. That'd probably be a nice way of saying this. So water evaporates out of the leaves, let's say stomata, due to heat from the sun. This lowers water potential. Yeah, it would. So this leads to tension. Excellent. So those of you who have said it, as soon as water leaves, it produces suction, right? And the word for suction is tension. So exactly the same way that when you like have a drink and you have a straw in it, as soon as you suck out some water from the top, it sort of produces this negative pressure on the inside where water wants to start moving upwards, right? So imagine you've got a little straw, right? As soon as the water which is here gets pulled out, then basically other water which is down here in your bottle wants to go and fill that space. You produce this negative pressure which the water in your bottle wants to go and fill. So you don't need to understand that very well, and it's actually really complicated. It's to do with like surface tension and a mess of stuff like that. But water evaporates out from the leaves due to the sun. This produces tension, negative pressure within the xylem. Okay, so there's now tension in the xylem. No teacher explained this like you. You're a best bio teacher. Thank you, nice one. I'm glad you're enjoying it. So this produces tension within the xylem, right? Why does that mean? Why does that mean that we end up having all of the water leaving together? So can you explain why? I understand why a few water molecules are going to go up. So the bit of tension is going to cause some water. Why do they all follow each other? Yeah, it does result in a transpiration pool. That's true. So this produces tension. Yeah, let's say that. So this produces like a transpiration pool. I like that. Whoops. Good. So hydrogen bonds cause the water molecules to stick together. So you guys are probably aware that you have hydrogen, oxygen, hydrogen. Oxygen is slightly negative, hydrogen is slightly positive. You end up with these weird little bonds that form. There you go. Right, called hydrogen bonds. And essentially, because of this interaction, because one is slightly negative and one is slightly positive, it means that they are slightly attracted towards one another. So this produces tension within the xylem. We can say hydrogen bonds lead to water molecules cohering together, or leads to water molecule cohesion. we can say therefore tension pulls up a continuous water column. That definitely does not look like how you spell that word. COLUMN. Apparently it is. Okay, so some of you might also want to talk about adhesion here as well. And the only reason I haven't said it yet is because not all specs use it. But if you've ever heard the word adhesion, you can add this in to get another mark. Essentially, the water molecules also stick to the xylem vessels. They stick to the cell wall of the xylem vessels, and that also allows them to move up, or it helps them to move up. I don't know, stops them falling down or something. But they interact with the xylem as well, and they can kind of stick to that, and that's to do with the capillary action, which Kang is talking about. So where do you mention adhesion? Just say that adhesion of the water molecule to the xylem vessel helps to produce this column of water or something. Is this called capillary action? Yes. Yeah, the sort of tension and evaporation is going to cause capillary action. So it says cohesion tension, so surely you can't talk about adhesion. No, that's because this is called the cohesion tension theory. Right, that's like saying, oh, well, it's called the evolutionary theory, therefore you can't talk about natural selection. No, it's just another element of it. Okay, that is all you need to say here. So that would get you all 3 marks. That is pretty much this in a nutshell. The only other thing that could be worth mentioning here is because it says how water moves up from the xylem, from the roots to the leaves, it doesn't actually ask you how it moves into the roots. That's the only thing this is missing. So if you were asked about that, you'd have to talk about osmosis going on for water to move into the roots. And for those of you that know about it, because some specs mention it and some don't, you would either talk about the apoplastic pathway where it moves through the cell wall, the symplastic pathway where it moves through the cytoplasm, or the vacuole pathway where it moves through vacuoles. Can you say capillary action? Yeah, Massimi, if you want to say capillary action, you knock yourself out. Would you need to describe how water moves through the leaf tissue? You could do a bit. It's just down a water potential gradient. Yeah, why not? Why not say that? So in order to get from the xylem, the vessel here, and the vessel here to the stomata, it's going to move down a water potential gradient. And again, it's going to either go through the cell wall or through the cytoplasm or through the vacuoles. Do I believe in macroevolution? Do I believe in the fact of evolution? Yes. Okay, so this question is relevant to some of you and not others. So if you've never seen this, then don't worry. AQA, for instance, don't worry about this. I think AQA are the only ones, actually. So plants transport water and assimilates through specialized tissues. Figure 4.1 shows a tissue plan of a vertical section through a part of a leaf. On figure 4.1, identify with the letter X the position of the xylem, and identify with the letter P the position of the phloem. And then can you name me the structure of R? Do I like Teletubbies? No, not so much. So yes, well done. R is indeed a vascular bundle. Well done for knowing that. Yeah, there is, for the person who asked me about evolution, there is as much evidence that evolution is true as there is that gravity is true. Right? I'm not going to jump out my window anytime soon. Just because it's a theory, it's not a law, because in science we don't really have laws. You only really do in maths. I'm going to believe in evolution because all of the evidence suggests it does. Right? If anyone had any evidence against evolution, they would have won the Nobel Prize. But there has never been like the wrong fossil found in the wrong place. Right? There's never been a bit of DNA sequence that doesn't kind of back up what we've already got to suggest. Like evolution is almost as certain, or not almost. Evolution is as certain as gravity is. Right? It's pretty definite. No, X is not the inside bit. Unlucky there. X is not the bottom semicircle either. So the xylem is on top. So xylem is on top, phloem is on the bottom. Okay, I don't know how you want to remember that. Maybe you guys are probably too young. There was like an operating system that computers used to have called Windows XP. Maybe think about that, X on top. Or you might think about your ex-boyfriend or your ex-girlfriend being on top of you and being a pain or something. I don't know, something like that. Your choice here, guys. But yeah, xylem is on top, phloem is on the bottom, right? In roots, you get this weird structure which looks like an X. Basically, the xylem is the X conveniently, the phloem's on the outside of it. Then stems, stems look a bit like this. You have like little circles like this. And basically the phloem's on the outside, the xylem's on the inside. So that would be phloem, that would be xylem. Okay, so you're expected to know that unless you do AQA. Yeah, it's your choice. That's how my brain would remember something stupid like that. Think of your ex being a pain somewhere and sort of hovering above you. There you go. Think of your ex-boyfriend or girlfriend just floating above you. You're like, go away. So the structure of cells is usually adapted to carry out their functions. The scientists use an electron microscope to look for further evidence to support the mechanism involved in the loading of sucrose into the sieve tubes. Suggest what evidence the scientists might expect to see in companion cells using an electron microscope. No, your girlfriend cannot be below you because otherwise you'll get that question wrong. She or he has to be floating above. Okay, good. Namrata, you're totally right. So suggest what you'd see if you looked at a companion cell. So you would expect many mitochondria for 1 mark. It doesn't say why, it doesn't say explain, it says suggest. So many mitochondria. Ribosomes to make co-transport proteins. See, I probably wouldn't say that, but you can say co-transport proteins. That's probably an easier option. So I wouldn't say ribosomes. Ribosomes tend to be quite small to see anyway. Would you be able to see a ribosome? You probably wouldn't be able to see a ribosome unless it was attached to the Golgi apparatus. No, endoplasmic reticulum. They're probably too small. But many mitochondria, definitely. Co-transport proteins. So like that. Oh, in fact, many ribosomes is given as a mark, so I'll give you that if you said that. You might see plasmodesmata, rough endoplasmic reticulum, something like that. You'll get a mark for that. Cytoplasm, no, that's not relevant. Why would having cytoplasm help you to be a companion cell? Nucleus, it does have a nucleus, but that doesn't help you to do your functions, so no. Parenchyma, that's a different type of cell, so no. Golgi, feasibly to make these proteins, yeah, but it's not given. So yeah, I'd go for one of the obvious ones. So mitochondria, definitely. Co-transport proteins, definitely. I'd say these ones are a little bit more tenuous, but you probably get a mark for them. Nucleus for ATP synthesis. The nucleus doesn't make ATP, so no. Mitochondria makes ATP. Oh, I love this question. This question comes up every year. You'll probably see this again at some point, and it's really easy. So a student used a potometer to measure the movement of water through a shoot of a plant. The potometer is shown in figure 1. As water is lost from a shoot, it is replaced by water from a capillary tube. So in one experiment, the air bubble moves 7.5 millimeters in 15 minutes. The diameter of the capillary tube was 1 millimeter. Calculate the rate of water uptake by the shoot in this experiment. Give your answer in millimeters cubed per hour. Show your working. How do I do it? Unlucky Jake. (15/60). Not quite sure why. So this is how many millimeters it moved in 15 minutes. So I like that you guys are starting to notice that you're going to have to times this by 4 at some point to turn that into 60 minutes. One thing to watch out for is that is millimeters. This is millimeters cubed. Ooh, 0.5 times pi times 7.5. Yeah, 0.5 squared times pi times 7.5. Very good. So if I'm working out the area, because in fact, I'm working out the volume, aren't I? If I'm working out the volume of this capillary tube, I first need to know what area it is. So the area of a circle, as you guys all know, because you've all done maths fairly recently, is pi r squared. So I know that pi is 3.14. R, my radius, well, it says the diameter is 1, so the radius has to be 0.5. So pi times 0.5 squared is going to be my area. I can't be bothered to actually write down what number that is because otherwise I'll get a rounding error. But if I then multiply that by the distance that it's moved, so it moved 7.5 millimeters. So let's say it moved from like here to here. That means that all of this has been moved in, hasn't it? So to work out my volume, I'm going to do my area multiplied by 7.5. And then finally, as you guys said earlier, I need to times it by 4 to make it per hour. So pi times 0.5 squared is that times by 7.5, that times by 4 should give you 23.56. You can't wait for my sales pitch. Oh, I haven't actually thought up what I'm going to say yet, so lower your expectations there, Amelia. So 23.6 is my final answer. What's the number of, how many decimal places / significant figures should I put out of interest? Did everyone get the maths here as well? Everyone happy with this? Yeah, I should really do 2. So the most that they've ever given me is 2. So 2, 2, 2. So let's do it to 2 significant figures. Let's go for 24. Yeah, you tend to copy what they've done. So the most they've ever given is 2, or you go one below what they've done. But I wouldn't say 20. I'd give 24. 20 isn't even given. So 24 is fine. 23.6 will get you the mark as well, to be fair. Why multiply it by 15? So they've told me that this happened in 15 minutes. I want it per hour. 15 times 4 is 60. When did I multiply it by 4? I did that first. So in my calculator, I would try and get in the habit of doing it as one equation. So what I did was I typed in pi times 0.5 squared. That was the first bit. And then it gave me that stupid number, which it tends to do. And then instead of writing what that actually is, I just said times 7.5, right, which gave me that horrible number. And then I wrote in times 4. And then finally, I had a look at my answer, and therefore I've had no rounding errors. Why did I multiply by 7.5? Because it moves 7.5. And if I want to work out the volume, I know the area is this circle. The volume is going to be that plus whatever distance is gone. So that's going to tell me the entire volume. Yes, the volume of the cylinder. Did I use 15? Yes, I times it by 4. Can you use a calculator in A-level? Yes. You'd be surprised, though. One of my most irritating students a couple of years ago, who I would love if he watched this, because he'd know exactly who I'm talking about. He forgot to bring a calculator to his A-level exam, his final year 13 A-level exam, and he had to come and get one off of the science department. Anyway, the list below describe 4 general features of leaves. From each list, select the leaf that belongs to a xerophyte. So place a tick in the correct box. First one has been done for you. So which is the mean number of stomata, would you guess? What leaf? So what do you know about xerophytes? What would you expect it to have in terms of stomata numbers? Yes, well done, guys. It's going to be F. So it's going to have few stomatas, so there is less water loss. Very good. Which is the next one? Surface area of one leaf. Yep, it's going to be G. It's going to be the one which has the least evaporation, so the smaller surface area. Thickness. Yeah, it's going to be K. Well done. It's the thickest one. That's going to have the least water loss. When it comes to xerophytes, right, 2 things that they do is they reduce evaporation. They reduce evaporation, or they reduce transpiration, right? So they do either of those 2 things. They either are really thick, so that water can't evaporate out. In fact, no, they're kind of the same thing, aren't they? So yeah, it's basically all about reducing transpiration, isn't it? So they're either having saturated water next to them, so that water can't osmose out. So I could say that. So they either prevent osmosis, or they prevent evaporation by having thicker cuticles and things like that. Anyway, so the transport system of multicellular plants consists of xylem and phloem tissues. The table below contrasts the structure of the roles of xylem and phloem. Complete the table using the most appropriate words. So xylem transports water and... No, not water. Minerals. Phloem transports are similar, such as... Not really glucose. Tends not to, yeah, it tends to be sucrose. I have no idea why, but glucose tends to get turned into sucrose. Sucrose or amino acids. Sieve tubes contain perforated cross walls. What's the equivalent for xylem? So phloem has cross walls, these sieve plates. What does xylem have? Dead lignin, liquefied, no end walls. That's what I'm looking for. So there's no end walls. It is literally a cell that used to look like this, which has ultimately had this bit in the middle break down, that bit at the end break down, that bit at the end break down. Xylem vessel walls are impregnated with, nice word, lignin. Well done. Xylem vessels contain. So there are many gaps in the cell walls. What are the gaps in the xylem vessels called? What is lignin? It's like a weird molecule which is waterproof. Bordered pits. Well done. Yeah, I'm going to go for pits. Pits are basically places where water can osmose in and out. So it means that water can get from one xylem vessel to another one. Not pores. They tend to be called pits. There are many gaps in the cell wall between companion cells and sieve tube elements. These are called plasmodesmata. I love that word. I don't know why I love that word so much, but I really do love that word. It makes me think of someone called Desmond or something. Desmata. That'd be a great name for a kid. Desmata. Come, Desmata. Anyway, describe the structure of the cell wall of a xylem vessel. So what is the cell wall like of a xylem vessel? What do you remember? This is a nice, easy question. Oh, glucose is too reactive. Jake, thank you for finding that out for me. So it contains lignin. Yes, I will give that. So yeah, lignin contains lignin. More basic than that. Cell wall's pretty dead anyway. It contains cellulose, yeah. So it's a cellulose cell wall. Well done. Yep, it's got pits. I'll give you that. Anything else? Hollow, that's not really a cell wall. A cell wall doesn't really matter if it's hollow or not. Dead cells, that's talking about the cell wall, not talking about the actual structure. So water and mineral ions, no nucleus. No, not really the cell wall. So we need one more thing. What does cellulose look like? Non-permeable. No, cell wall's partially permeable, a little bit. I see what you mean. It's not the most. What could another word be for, what could you say about the cellulose? Think back to like really early biology. Good. Microfibrils, I'll give that. So cellulose containing microfibrils. Think back to easier things. Yeah, microfibrils, beta glucose you could talk about. Don't know why I wrote it like that. So beta glucose, you could say, what else? What does permeable mean? It means things can be through it. We could say that there are like 1,4 glycosidic bonds or something like that. Anything like that. So beta glucose, microfibrils. You got marks for saying that the lignin is spiraled. You could also say that there is something called pectin and hemicellulose in there too, which are basically strengthening. Right, any of those things you can get marks for. I imagine the person in question probably doesn't have much better to do with their life, if I'm totally honest. So the stem of plants contains xylem vessels and sclerenchyma fibers. Compare the functions of xylem vessels and the functions of sclerenchyma fibers. Would you get 4 marks with only 4 features? Yes. So compare the function of a xylem vessel and a sclerenchyma fiber. So some of you will have never come across sclerenchyma before. So it's like, oh, I don't want to say, I'll give you away the mark if I tell you what it is. But it's a fiber which you get next to, not AQA, no. Fiber that you get next to the xylem. Can you guess what it does? Like, what could a long fiber be doing? No, not AQA. OCR, yes, I believe OCR. What could a really long fiber be doing in a plant? You haven't seen it in OCR? Okay, I might be wrong. Not waterproofing, good guess. Yep, just supporting. So it just gives strength. So sclerenchyma provides strength. This question's a bit of a weird one. It's used for support strengthening. Does a xylem do that? Yeah. So we want to say that both the xylem vessels and the sclerenchyma fibers provide strength. So that's my first mark. So the xylem and the sclerenchyma fibers provide strength. What is something that only the xylem does? They are not the same, Mimika. They're similar, not the same. Good. So xylem carries water. What else does the xylem do? So carries water, provides some strength, and let's make sure we get this mark by writing both. Good. It also transports mineral ions. To be fair, I probably should say transports here as well. Transports mineral ions. Easy. Right. Okay, cool. That's something which is a bit of a weird one, which possibly you might not have heard of. I thought OCR was in, I thought sclerenchyma was mentioned in OCR, but there's enough of you saying that maybe there isn't, that maybe there isn't. I don't know. Maybe it isn't. If I'm totally honest, I could have forgotten. I'm pretty sure LXOA have it. Is there any difference between OCR? Yes, there are differences in content. Like, not a lot, but little bits. Right, okay, let's look at one last question. I like this question. So really, really, really applied question just to finish off then. We've got a couple of them. So under the correct conditions, new roots grow from a cut end of a plant stem. Scientists investigated the effect of substance X on the growth of new roots. She used a ringing experiment to investigate the movement of substance X in stems taken from lemon plants. She cut out a length of a stem from each plant. She then put a small block of agar on top of each length, and some of the agar blocks contain substance X. Diagram shows the treatments, blah, 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 blah. So X is just agar, X is agar and substance X, F is substance X, agar, and the stem has been ringed, and substance X has been cooled, and it's intact, and it's got substance X. The table shows their results. So she grew lengths of stems, for 6 weeks. She found a number of roots per length of stem. They grew at the other end, so they grow down here. Blah, 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 blah. She has 4 treatments. Here are the number of stems, the number of roots per stem. Okay, so question 1. Treatment D is a control. Explain how the measurement obtained from this control is used by a scientist. So, yeah, what's the point in doing a control? Yeah, this is a recent question, this one. So what's the point of a control? Cambridge International is another spec, yeah. Good. It's a comparison, but comparison what? For what? In fact, let's say that. So it allows comparison. To be made. So you have your control, so you can be like, oh, well, look at E, compared to that, it's more. Look at F, compared to that, it's about the same. Look at G, it's less, right? So there's that. So you can compare. What does it also let you find out? No, nothing to do with validity, nothing to do with anomalies. So it allows comparisons. In this experiment in particular, what does it tell me the effect of? Whether ringing has an effect? No, it's not whether ringing has an effect. There's only one thing which is different in that control to all the other ones. Good, it's the effect of X. So it allows comparisons to be made and shows us the effect of substance X. Yeah, so show me what substance X is doing, and it allows me to therefore make comparisons. Okay, use the diagram and table above. What can you conclude about the treatment D and treatment E about root growth? So what does D and E tell us about root growth? I think this was taken from AQA, if I remember correctly. Doesn't mean it's not relevant to the rest of you. It is. So what does this tell us about root development? So go on, X. X does what to root growth? Yeah, I think it is AQA. Ooh, Jess Smith, well done. X doesn't actually cause root growth. So D doesn't actually have any of substance X at all, and yet I still got some root growth. So actually you can argue the opposite. You can say X isn't actually needed for root growth because I got growth without it. I'm really impressed someone actually worked that one out. Most people miss that. So X isn't actually needed for root growth because without X, I still have it. However, X does seem to increase root growth. Definitely. What else does this tell us? It causes faster mitosis. We don't know if it causes more mitosis. This question is confusing. Agreed. So what does D and E show us? The last mark I actually don't think you'll get. The last mark's a really tough one. So the only other thing it shows us is if substance X is put at this end, if it causes growth down here, what does that tell you about what's happening to X? The only way to grow is by mitosis. Yeah, that is true. That is true. Feasibly could be doing meiosis, but yeah, you're right. It almost certainly is mitosis. Good. This D and E also show that X is transported down plants. I think that's a horrible mark, that last one. Yeah, so it's transported down plants through the stem. In fact, no, that's enough to get the mark, actually. Right, final question, I believe. So the mass flow hypothesis is used to explain the movement of substances through the phloem. Evaluate whether the information from this investigation supports this hypothesis. Do not consider statistical analysis in your answer. So, go on, I'll go back. For those of you that were stuck there. So X, it shows us that X isn't needed because I still get some growth without it. It shows that if you do have X, then you get more growth. But it also shows that X must be able to move because it's causing growth elsewhere. So X must be able to move somehow, right? This one, basically evaluates probably the hardest questions. Evaluate whether this information from the investigation supports the hypothesis that that substances move by mass flow. So does this support that hypothesis? Good. So there is clearly a sink and a source. So in this, evidence that backs it up is there is a source of substance X at the top. I never thought anyone would get that mark. So source of substance X at the top. and there is a sink at the bottom. That will get you a mark, right? Anything that you can take that is similar will probably get you some marks. So why is growth at the opposite end? Why does that tell you that this is using mass flow? If you can tell me why. So what does it mean? If it starts here and it moves all the way down here, why is that suggesting that this is mass flow? What did mass flow use in plants? Just go back to that like those first long questions we went through. What does that mean it's using? Oh, I'm getting properly hot in this room. Ugh. Doesn't necessarily that so it well, I'll get to ATP in a second, maybe. I think you are late. Phloem, yeah, Talia, if you can say that essentially, because it's moving, it's probably moving through a phloem, that is evidence that it's using mass flow. So F, in fact, yeah, F tells us that the phloem is involved. So if we say F shows the phloem is involved, because I basically get no additional growth if I take my phloem away. So F shows that phloem is involved. And I know that that is used in mass flow. So that's a good mark. What does this one show us? If it gets cooled down to 4 degrees and I get less, why does that tell us that this is using mass flow? You don't get a second point. So why is this evidence of mass flow? Well, mass flow uses a phloem. And when I take the phloem away, I don't get very much growth again. So that's evidence that this is using mass flow. Sorry. Why is it slowed down if it's cooled? What bit of translocation doesn't work as well when it's cold? Shows chemical reactions are involved as and temperature is going to decrease the rate of transport. Yeah, very good. So this is showing me then that when it's cool, so when cooled down, so G shows me, you can get 2 marks for this. G shows that metabolic reactions are involved. By metabolic, I mean respiration. So G shows that metabolic reactions are involved. Respiration, because when it doesn't happen, because when it is cooled, the growth reduces. Okay, so essentially, we know that active transport is used to get all this sucrose into the phloem. So I'm assuming that substance X is going to be needed to be actively transported in. If it's cold, aerobic respiration is going to happen slower, and therefore you're going to get less growth. So there's 3 marks. One last mark then, and this is horrible. So this last mark is very, very hard to get. So I'll be impressed if anyone can get it. In fact, no, I might have to tell you. Less kinetic energy, fewer particles of correct. No. So you can get marks if you say that the evidence against, so evaluate, you tend to have to say something for and against. Right? Evidence against is if it did use translocation, then you tend to get a bulge wherever you've cut the stem. So if you cut the stem, you tend to get a bulge which fills up with loads of sugar. And this experiment doesn't say that that's happened. So that is evidence against. So no bulge above the cut. I don't know how anyone would get that mark. I think that's a crazy mark to get. You'd get a mark if you said that there is no role, but like we don't know how water's involved. So there's no role for water in this. So how can you have the pressure gradient produced? I think that's a bit of a tenuous mark as well, if I'm totally honest. You get a mark if you say that movement could just be due to gravity. That's an easy mark. So we don't know from this that it's not just gravity pulling X down, with assuming that it's not gravity. You can get a mark for saying the roots still grow without the phloem, and you get a mark for saying that there are no leaves or sugars or photosynthesis like causing anything to be produced. I think that's a really stupid question. What does a bulge mean? No, imagine if my head just started getting really fat all of a sudden. My head would be bulging. Don't know why that's the first thing that popped into my head. But yeah, so evidence against mass flow, that's the main one. There's no bulge. You'd expect to have a bulge. Right, I just thought of something good. So what's the link between a xerophyte and snap revise? Who can tell me? Oh, because if you took away the phloem, right? Essentially what's going on, everyone else think about that question. If you took away the phloem, then all of the sugar, which is like moving down here, it can't move any further. So it can't move through this bit. So all of this sugar is just going to build up here. You end up getting like massive bulges that start to form. So if you Google ringing experiment, Google that, right? And you'll see a whole load of trees turn up, and they always have a bulge just above where you've cut them because it's filled up with sugar. Anyway, what's the similarity between a xerophyte and snap revise? You guys know me far too well. Yeah. So a xerophyte, oh, you can't really see this. A xerophyte reduces water loss. However, snap revise doesn't reduce water loss. It reduces knowledge loss. Yeah, I came up with that one in about 2 seconds. Love it. Right. So just like a xerophyte, where you find them, oh, this gets even better. Xerophytes commonly are found in saline conditions. They are found in hostile, extreme conditions, cold conditions quite often. And they tend to somehow survive. Despite all of this horrible stuff going on around them, they tend to survive. And snap revise is very much like that in my mind. And when I applied for this job, I was thinking, oh, how is snap revise like a xerophyte? And then it just came to me. So snap revise is like a xerophyte because it will reduce any knowledge loss, right? Okay, I can't say that because that would probably be false advertising because you might lose some knowledge. But I can say that it's very much acting in the same way. So a xerophyte still has evaporation of water out of it. It's just less. Snap revise, you will get some knowledge loss, but it'll be much, much, much less. Dad joke 101. Yes, exactly. The weird thing is I'm not even a dad yet. I feel like maybe I've somehow inherited something horrible from my father. But anyway, here is snap revise for those of you that want to see it who haven't seen it before. Essentially how it works is let's pick something. Let's go to one I don't normally pick on. Let's go to, there you go, Edexcel biology, right? Essentially what you have is you have a means to learn in a way which will hopefully help you quite a lot more than just sitting with a textbook. So for instance, if you were wanting to revise epigenetics and cancer, you could click on the bottom that says epigenetics and cancer and fats. I'm not going to go on that one. I'm going to decide against that because that one's a bit rubbish in terms of the amount of resources I have for it. Let's go for, let's try some xylem and phloem. That makes sense. Oh, that one's a bit rubbish too. No, I'm going to sack that one off too. Hang on. Third time's a charm though, eh? Third time's a charm. Edexcel clearly doesn't like me. Let's go for introduction of biological molecules. Essentially, what will happen is, I've chosen a terrible one, is you will essentially get given a test. So when you sign up, it will do a little test on the topic that you're doing. You can go through the tests and you can be like, oh, I clearly don't know what I'm talking about here, right? So after my 4 questions, which I purposely got all of them wrong for, it will tell me if I got 0 correct. I clearly don't know the biochemical basis of life. I clearly don't know what biological molecules are. I clearly don't know what monomers and polymers are. And I clearly don't know what a condensation or hydrolysis reaction is. So when I click get started, everything that I didn't know, it will put in orange. It will load up all the videos in orange, so I know which ones to watch. And say I've watched this video and I haven't got it, I can write a comment being like, I thought that this was like a xerophyte, because that's how people spell in my head. And I wouldn't lose knowledge. Help. You could write that. And essentially what will happen is I'll get a nice little message. It will pop up on my screen and I'll see your message and I can then start responding to it, right? I can write you a nice little comment being like, oh, no, don't worry about it. I'm here to help you, right? Essentially, when you've done that, there'll be a quiz that you can have a go at doing where you can see how you're doing after going through like however long that was, 16 minutes of videos. And then when you've done that, you could go through an exam question where someone will go through an exam question with you and essentially you'll be able to see if you can do the thing that you're supposed to be able to do. Now, quite bizarrely, I seem to have chosen some that don't have an exam pack that go along with them, which is very bizarre because I know that almost all of them do, but this one seems to be lacking some. Oh, here are some of them. So you can look at the spec points as well if you wanted to. But after that, if you still didn't have enough of a means of checking if you knew stuff, you hadn't assessed yourself enough, you could do any one of these questions. Describe the structure of starch, click on the answer, there is your answer, right? So there are literally loads of these. For every single topic in your specification, there'll be some questions about it. We also have, and we don't for this bit of LXLA, but we also have like a revision guide that you can download as well. Let's say that wasn't enough. Let's say that you were still thinking, oh God, this still really sucks and I don't know anything. There are also web classes. So I don't just do these YouTube classes, I do web classes literally every day. I haven't actually scheduled any for next week because I've been lazy this week. But I did one earlier today about maths. I did one yesterday about antibiotics. I did one the day before that about circulation, clotting, CVD in the liver. And basically I do them every day apart from Friday. So I've been doing loads of these sessions. I've been doing loads of drop-ins. I'm doing a book club tomorrow. Here are my drop-ins that I've done the last few weeks, so you can see how many there are. I basically just do drop-ins and I do lessons. And if you don't know something, you can just be like, oh, I don't get this. Do you mind going through it? And I'll say, yes, that's fine. And basically, that is my job. That's what I do here at Snap Revise. And it's a really, really good way of learning. Like, I know I try and make jokes about it, but genuinely, I don't know, for not that much money, and I know it costs some money because obviously I need to be able to afford my roof, you get a lot of lessons, right? So when I used to be a tutor, I used to charge about 40 quid per session, and now you pay about 50 quid a month and you'll get, I don't know, at least 10 lessons that you would go to, probably more than that. So I think last week I did about 48 lessons per month. I think it's a bit fewer now that people are going to start going back to school, but you'll get loads of lessons basically to go over things. So if you aren't convinced, feel free to set reminders. You can carry on coming to these YouTube sessions. But if you are interested, then have a look at this. Essentially, I've got £10 off that you can use. I've also got this Instagram thing where I've got one favorite at the moment, but if you post an Insta story, if it basically makes me laugh and makes me happy, I might choose you to have a free account, which is worth about 200 quid up until September. Okay, so, happy National Teacher Day. I didn't know that was a thing. Guys, I'll read your messages, but otherwise I will stick around and see if you have any questions. What are we saying? My tablet isn't powerful enough to run Snap Revise. That is shocking. That's really weird. What tablet do you have? Am I ready to be a dad? I am not ready to be a dad, no. My brother, on the other hand, well, he is ready to be a dad, and recently my sister-in-law got pregnant, so I'll be an uncle soon, so I can have uncle jokes. Is there a geography one? I don't think there is a geography one. I think there's one for GCSE, but we've got biology, maths, chemistry, physics, and that's it. They're the big boys. How much is a subscription? So the cheapest subscription is about £10 a month. The most expensive one's about £50 a month. Will you and your pals at Snap Revise do English Home Study Club stream soon? I don't know. I don't think so, because I don't think we have anyone employed who does English. How many people participated in the last competition? Oh, I reckon I had about 6 or 7. Yeah, no, I put them all on my phone, actually. Hang on. I don't know if I deleted them. Just to prove to you that this is actually me who looks at these. Hang on. I know some of you won't believe me, although I might have actually deleted them since then. Oh, no. Here you go, look. Someone made that. That was a good one. That's probably my favourite one so far. I like this one as well. Hang on, where is it? This one is also a worthy contender. But yeah, I got 1, 2, 3, 4, 5, 6, 7, 8. I got 8 this week. So there is a 1 in 8 chance at the moment of you winning. What else are we saying? Can you do another live on here on Tuesday because of the bank holiday? No, I'm not doing another bit of work. Someone else does one, I think, on Tuesday. I think George does a chemistry one. What should year 11s do during holidays? Year 11s need to make sure they actually know what they're getting their grades for in science, right? So thank you for congrats on being an uncle. I don't think I did very much. If you're in year 11, which I doubt many of you are, basically just learn all of GCSE properly, right? Don't go into year 12 being like, oh, I'll just get by on what I know now. Learn the GCSE properly, right? Properly learn it. Snap Revise could be the cure to COVID-19. It could be, although that's actually definitely not true, and I don't want you to take me down. YouTube, I know it's not true. Snap Revise won't cure it. The bite-sized videos, that's another thing. They're very laggy on your tablet. That is annoying. Who won last time? Someone, I can't remember his name. Sard, a guy called Sard, he won last time. Right, anyway, I am probably going to head off then if no one else has any other questions. So have a lovely rest of your day, guys, and have a lovely weekend, bank holiday, and I'll catch you again in about a week. Right, see you later, guys. Have a lovely afternoon. Bye.